Hello students, so we are studying our first lesson, that is reproduction in lower and higher plants. Okay, so in the last period we have studied double fertilization. Okay, so today we will study development of endosperm and then development of embryo. Okay, so let us study first of all endosperm. Okay, so as we have studied in double fertilization, there is a two types of fertilization. Okay, two times there is a fusion of male and female gamete. Okay, so when then we have studied the formation of triploid primary endosperm nucleus in the last lecture. Okay, so from that there is a development of endosperm. So endosperm develop from pen or primary endosperm nucleus. And what is the function of endosperm? Endosperm will feed the germinating seed, okay, or it will feed the germinating embryo which will develop later on in the new plant. Okay, so let us study the triploid primary endosperm nucleus repeatedly divides mitotically to form nutritive tissue called as the endosperm. So by the mitotic division, pen will develop into endosperm, which is a nutriting or feeding tissue. In post fertilization changes within the ovule, the embryo and endosperm are seen to develop simultaneously. So after fertilization, simultaneously means at a time there will be development as well as development of endosperm as well as development of embryo will further develop into seed, and afterward germination a new plant will form. And what is the function of endosperm will feed that growing embryo. Okay, the other cells of embryo sac disorganize sooner or later. So all other cells disorganize or they become extinct. The formation of triploid endosperm nucleus triggers cell division which leads to formation of endosperm. So from this pen or primary endosperm nucleus there will be a development of endosperm. So here diagram is shown how this endosperm is formed. Okay, then there are three types of endosperm. First one is a nuclear endosperm, second is cellular, and third one is halobial. So in your textbook the diagram is shown. So let us see one by one. First one, nuclear type. Okay, so it is the most common type found in the 161 angiospermic families. Angiospermic means which flowering plants. Here the primary endosperm nucleus repeatedly divides mitotically without wall formation to produce large number of free nucleus. So as shown in the diagram, first of all nucleus will divide again and again. Many nuclei are formed here in the second figure, and then around this nucleus there will be formation of cell wall, and structure will look like the third figure. Understood? So what is the characteristics of nuclear type? There will be, first of all, there is a division of nucleus to form many nuclei, and then cell wall will be formed around these nuclei. Okay, so that is the nuclear type of endosperm. A big central vacuole appears in the center of cell, pushing the nuclei toward the periphery, means toward the side of that circle. Later walls develop between the nuclei, hence multicellular endosperm is formed. So many cells are formed, but in several cases cell wall formation remains incomplete. Example, wheat, sunflower and coconut. So cell wall is not formed around all the nuclei, it is incomplete. Coconut has multicellular endosperm in the outer part and free nuclear as well as evacuated endosperm in the center. Okay, so what is the characteristic of this nuclear type of endosperm? What is the important point? The primary endosperm nucleus repeatedly divides mitotically, means many times there is a division of nucleus without wall formation, means in the first stage wall is not present. Afterward, when many free nuclei are formed, there is a formation of cell wall around these nuclei. Okay, that is nuclear. Then second type is cellular type. Okay, so in some plant, division of triploid primary endospermic nucleus is immediately followed by wall formation. So there is a division of this nucleus or primary endosperm nucleus take place, and immediately after the formation of nucleus there is a wall formation, that is called as the cellular type of endosperm. Here diagram is shown. Okay, oospore, and that is pen. Then from that many nuclear form, and immediately after the formation of nuclei cell wall is also formed, that is cellular. And then C, halobial. Okay, so it occurs in order halobials of the monocots. So from the monocot plants there is one order halobials. In that case this halobial type of endosperm is present. In this case first division of primary endosperm nucleus is followed by a transverse wall which divide the cell unequally. So here you can see halobial type. Here a transverse or horizontal wall is formed. Okay, the smaller cell is called as chalazal cell. The smaller one is called as chalazal cell, and the bigger one is called as, or larger cell is called as micropylar cell. Then the nuclei in each cell divide by free nuclear divisions, and then wall develop between nuclei in micropylar chamber. So here you can see after the formation of two cells there is a division of nucleus, and then many nuclei are formed around which cell wall will be formed. Okay, then it is intermediate between cellular and nuclear type. So it is a middle type of endosperm between nuclear and cellular. Okay, then mosaic endosperm. So in some plant like corn, it contains the patches of two different colors, that's why it is called as the mosaic pattern. Okay, then so this is about the development of development and types of endosperm. Then development of embryo. Okay, so as we have seen in the double fertilization, two times fertilization take place and there is a formation of zygote. Okay, so embryo means after formation of zygote, the zygote will undergo some changes and it will convert into embryo. So what is embryo? Embryo is it means it will convert into seed. Okay, or seed like structure which will further develop into plant. Okay, so all the plant part like root, stem, leaves, they are inside the embryo, and the conditions are favorable, means temperature, water, sunlight, that embryo will start developing and it will develop into new plant. Understood? Embryo and endosperm means it is a feeding or nutrition, means endosperm gives nutrition or food to the developing embryo. Embryo will further develop into new plant. Okay, so let us see. So there are many steps in this development of embryo also. So let us see one by one. The process of development of zygote into embryo is called as embryogenesis. So there are several steps in the development of this embryo. This zygote will convert into embryo by the process of embryogenesis. The embryo is developed at the micropyle end of the embryo sac. So this we have studied in the earlier lectures. This embryo develops to a micropyle end where there is a egg cell. Okay, and egg cell and one male gamete, that will form zygote, and zygote further develop into embryo. The growth of embryo triggers only after certain amount of endosperm is formed, because endosperm is a food of this embryo. So unless and until food is not there, embryo cannot develop further. So when there will be certain amount of endosperm will be formed, after that automatically embryo will start growing as it will get its food. Okay, then after fertilization the embryonic development begins. So after fertilization zygote will form, and from zygote this embryo will develop. The zygote divides to form two-celled embryo. The larger cell toward the micropyle is called as basal or suspensory initial cell, and smaller cell toward chalazal end is called as terminal or embryonal initial cells. Okay, so the larger cell, here it is shown in the diagram, the larger one which is toward the micropyle end, that is called as suspensor initial, and the smaller which is toward the chalazal end, that is called as the embryonal initial. So two cells are formed in the first step. The suspensor cell divides transversely in one plane to produce filamentous suspensor of 6 to 10 cells. So as shown here, there will be division of this suspensor cell, and there will be formation of filamentous 6 to 10 suspensor cells. Then the first cell of the suspensor, the first cell of the suspensor toward the micropyle end becomes swollen and function as a haustorium. So what is mean by haustorium? So you have studied haustorium means haustorium will function, it will take and transport the nutrients. Okay, so that is the function of this haustorium. So the first cell of the suspensor toward micropyle end become swollen, become big, and it will function as a haustorium, means it will take and transport the nutrients toward this growing embryo. The lowermost cell of the suspensor is known as a hypophysis. The suspensor help in pushing the embryo in the endosperm. So what is the function of this what is the function of this suspensor cell? So this suspensor will help to push the embryo in the endosperm. And what is the function of endosperm? Endosperm will give food to the developing embryo, which is going to become plant afterward. Then what is the function of hypophysis? So hypophysis will later on develop into root. Okay, the embryonal initial undergoes 3 successive mitotic divisions to form octant. Octant means 8 cells. Okay, so there will be formation of 8 cells by the mitotic division. The planes of division are at right angle to each other. So here the diagram is shown. The lower tier of the 4 cells of the octant give rise to hypocotyl and radicle, whereas the 4 cells of tier form the plumule and the 1 or 2 cotyledons. So 2 cells are there. So from 4 cells there will be upper 4 cells will be give rise to plumule. Okay, so plumule means the part which will grow into stem or shoot. Okay, so upper 4 cells will develop into plumule. Plumule will afterward plumule will develop as a stem. Initially plumule comes out of the soil on above the ground. It will develop some small leaves, and afterward later on it will develop as a stem. Cotyledons will be developed along with the plumule, and the lower tier of the 4 cells which gives rise to hypocotyl and radicle. Okay, so radicle, you know that the part of the developing embryo under the soil which will develop into root is called as radicle. Understood? So out of 8 cell, upper 4 cell will be grow above the soil to form stem, leaves, etc., and the lower 4 cells will develop into radicle, which will further develop into root and root tip. Okay, then the hypophysis by further division gives rise to part of radicle and root cap. So root and root caps, radicle will give rise to roots. Subsequently, the cells in the upper tier of octant divide in several planes so as to become heart shaped, which then forms 2 lateral cotyledons. So subsequently, afterward, the upper tier of 4 cells will develop, it will become heart shape, and then it will form 2 cotyledons. Okay, and the terminal plumule. Okay, plumule will develop into stem. Further enlargement of hypocotyl and cotyledons result in curvature of embryo and appears horseshoe shape. So the developed this one will look like a horseshoe shape. Okay, then the embryo development is similar in both dicot and monocot. Dicot means having two cotyledon. Di means always two, and monocot means having single cotyledon. For example, wheat, jawar, they are monocot, while as groundnut, red gram, they are dicot. Okay, the difference appear later. In monocot embryo, single cotyledon occupies terminal position and plumule is lateral. Plumule means growing shoot or stem. The single shield shaped cotyledon is called as scutellum. Okay, so as there is a single cotyledon and it is shield shaped, which is called as scutellum. The protective sheath of plumule is called as coleoptile. So there is a protective sheath around the plumule. Plumule means which is going to develop as a stem. So protective sheath is called as coleoptile, and that of radicle is coleorhiza. So there is radicle means from which roots are going to develop. So it has also protective sheath which is called as coleorhiza. Finally, ovule is transformed into the seed and ovary into fruit. So finally, after this all cell division and growth, ovule will transform into seed and ovary will develop into fruit. So in this way there is a development of embryo and endosperm. So let us revise again. First of all, development of endosperm. So endosperm is a nutritive tissue or which give food to the developing embryo. Okay, then it develops from the pen or primary endosperm nucleus, which has formed in the double fertilization. Okay, then there are three types. First one nuclear, second one cellular, third one halobial. Okay, so in nuclear type of endosperm, there is a formation of many nuclei first of all in the first step, and afterward there is a formation of cell wall around these nuclei. Okay, then the second type is cellular type. In cellular type there is a formation of nucleus will get divided into many nuclei, and then there will be formation of cell wall around these nuclei. That is called as cellular type of endosperm. And last type is cellular. So in cellular, as shown in the diagram, third one is halobial. So in halobial, here first of all two cells will be formed. Okay, and in these two cells there will be many nuclei will form, and then around these nuclei cell walls will be formed. Okay, so that is about the endosperm. Then next one is development of embryo. So embryo is, what is meant by embryo? After fertilization there is formation of zygote, that is from male and female gamete will form zygote in sexual reproduction, and then that zygote will develop into embryo. Okay, and in embryo there is, it is a all plant parts are there inside the embryo, and when the conditions are favorable, that embryo will develop into new plant. Okay, so here there are steps in this development of embryo. So what is the zygote divide to form two-celled embryo, which is called as proembryo. The larger cell toward the micropyle is called as basal or suspensor initial, and embryonal initial. Embryonal initial is toward the chalazaend. So it is the first step for which is called as proembryo stage is two-celled. Okay, two cells are developed. Then from suspensor, what is the function of suspensor? It will push the embryo into this one. It will push the embryo into endosperm for getting nutrition. Okay, then the first cell of the suspensor will function as a hostorium. Hostorium means it will take and transport the nutrients toward the this embryo, developing embryo. Then the lowermost cell of the suspensor is known as hypophysis. Okay, and this hypophysis will afterward develop into root or root cap. The suspensor helps in pushing the embryo in the endosperm. So the function of suspensor is to push the developing embryo into endosperm to get the nutrition. Then the embryonal initial undergoes 3 successive mitotic division to form octant, means 8 cells are formed. The divisions, the planes of division are at right angle to each other. The lower tier of 4 cells of the octant gives to hypocotyl and radical. Those lower 4 cells will develop into hypocotyl and radical. Radical means which will develop into root, and upper tier or upper 4 cells will develop into plumule and cotyledons. Plumule will develop into stem. The hypophysis by further divisions gives rise to the part of radical and root cap. Radical means from which roots are developed. Subsequently, the cells in the upper tier of the octant divides in several planes so as to become heart shaped. So they become heart shape, and which form 2 lateral cotyledons. Okay, so 2 cotyledons are formed and a terminal plumule is formed from which stem will develop further. Further enlargement of hypocotyl and cotyledons result in the curvature of embryo and it appears horseshoe shape, which will further develop into the new plant. The embryo development is similar in both dicot and monocot. The difference appear later. In monocot, embryo single cotyledon occupies terminal position, because in monocot there is only one cotyledon, and a plumule is lateral. Plumule means stem, which will develop into stem. Plumule will develop into stem. The single shield shaped cotyledon is called as scutellum, and the protective sheath of plumule is called as coleptile, and that the protective sheath of radical is called as coleorhiza. Okay, so finally ovule will be transferred into seed and ovary will develop into fruit. Okay, so diagrams are given. You should go through these diagrams. Okay, thank you.